This mini lecture is to go back over bisulfite sequencing and ChIP-seq techniques. So the purpose of bisulfite sequencing is to determine which cytosines, Cs in a DNA strand or DNA sequence are methylated. So, remember that DNA methylation is an epigenetic mark, and it's associated with gene off. So if you're trying to determine if the sequence or the gene you're looking at has DNA methylation, you can't just do regular DNA sequencing because it will just tell you the A, G, Cs, and Ts. It won't tell you methylation. So in order to determine which Cs are methylated, you have to treat the DNA with bisulfite, okay, which is a compound that will eventually deaminate. So here's our cytosine. It will remove this amine group. It's replaced with an oxygen. Okay. You have a hydrogen here, and the double bonds are moved. But what the result of this chemical treatment is a nucleotide that looks like uracil. Okay. So remember, uracil is not normally found in DNA, but it does base pair with adenine. So U and A's can base pair in RNA. So this nucleotide is semi-stable, but it's going to change your DNA sequence. Okay. If the cytosine has a methyl group attached to it, then bisulfite does not convert it to a uracil and it remains intact. So the key here is a cytosine will become a uracil, which will become a T in the final DNA sequence. And a methylated cytosine will stay a cytosine after bisulfite sequencing. So let's look at an example. Okay. Here's your original sequence up top. Here's your bisulfite converted sequence. Okay. We are always writing the sequence 5' to 3'. Okay, that's how I will always do it unless I mark it differently. Okay, so if there's no 5' to 3', you can safely assume that it's going 5' to 3' left to right. Okay, and our rule is that a 5' C next to a G at the 3' end will get methylated, but that's not always the case. So you can see here that this cytosine and this cytosine and this cytosine and this cytosine, even though it's a CpG cytosine, they were not methylated because in the bisulfite converted sequence they became Ts. And the same for that cytosine. Okay. And the same for this cytosine. But when we come down here, we see this cytosine and this cytosine remain in the sequence. So they're in the original sequence and they're in the bisulfite sequence, which means that these guys were the ones that were methylated. Okay. So not all CpGs are methylated. If you look at some other sequences, sometimes different Cs are methylated, but our rule is to look for those CpGs, but you never know until you do bisulfite sequencing. The Cs that remain were methylated. Okay, so let me show you why the Cs are converted to Ts. Okay, again, all I've done is split that diagram. So here's our original sequence, 5' to 3'. We add bisulfite, and all these C's now look like U's. Except for, let me remind myself that these two were methylated. So I'm just copying this sequence, but wherever there was a C that was not methylated, it now is going to look like U. Okay. It's supposed to be a G. Alright, so this is still 5' to 3'. We've just chemically changed this DNA strand. Okay. When you make the complementary strand, so when DNA replication happens, I'll just change colors here. It's going to be anti-parallel. We're going to learn this all this coming week. U's will base pair with A's. G's base pair with C's. U and A and G and C and A base pairs with T. C, G, C, G, C, C. Okay. I want you to notice this is the complementary strand. It's anti-parallel. And we're always going to compare the same strands. So what I really need is the 5' to 3' strand. Okay. So again, if we replicated this purple strand, we would get T, 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 A's and T's base pair, G, 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 A, T, G, G, A, G, T, T, G, C, we have a C, G, 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 C, G, G. And if you line it up, these are the same sequences. So the shortcut is that we don't write the bisulfite in the complementary, but you can do that, right? You can do that to figure this out. Okay. What we're jumping to, the idea is that you understand any unmethylated C's are going to become T's. And they become that because first they look like U's, which base pair with A's, and when that strand is copied back to the 5' to 3' orientation, A's base pair with T's. Okay, so I hope that helps you with bisulfite. ChIP and ChIP-seq. So ChIP stands for chromatin immunoprecipitation. Chromatin, remember, is our DNA plus histones. Immunoprecipitation means it uses antibodies to pull out of solution or pull down whatever is bound. Alright, so let's take a look at this, oh darn it, technique. Okay, so here we have chromatin, right? DNA wrapped around our histone proteins. And either with an enzyme or with sheer force of sonication, we want to break these into chromatin, into small units. Because you don't want to pull down a huge chunk of DNA that's not specific. You want to look for very specific interactions. Once you have this broken down, you add an antibody shown here in blue and red that's specific to histone. Antibodies specific to histone of non-histone proteins are added. Okay, I should have looked at this more carefully. Antibodies that are specific to histone, modifications. Okay, so for instance, maybe we want to pull down H3K9 methylation. You can have an antibody very specific to that, and this is supposed to be the little histone tail, and if that modification is there, the antibodies bind, and the antibodies will have some kind of bead or magnet on them, and you will pull them out of solution. Okay. Then you get rid of, so what I forgot to say is that you're cross-linking the proteins and the DNA up here in the first step. That just keeps the reaction so they don't fall apart. Then you reverse the cross-links and you get rid of the protein, so that you can analyze the DNA. And so there's a couple ways to do it. You can analyze the DNA by running blots, so that would be the ChIP, or you can add sequencing, so you can get the AGCT actual sequence that's interacted with that histone, and that's ChIP-seq. So they're basically the same thing, it's just what are you doing with the DNA. So let's look at a different picture. This is showing the same thing. Okay, what it's showing you is up here you have histone modifications. Okay. You isolate the chromatin from the nucleus. Remember, this is DNA plus histones. You break it into pieces. You add the antibody that's specific for what you're looking at. Okay, so it could be H3K9 methyl 2, H3K27 methyl 3, acetylated H3, whatever it is. This will pull out these specific complexes. So I want you to see you lose all these other ones that are not with that modification of interest. You get rid of the proteins so that we can analyze the DNA, and this is showing PCR or sequencing so that you can figure out what DNA you're looking with. So the type of data I would give you is something like this that we worked on in class. So these, nope, not that. These are the specific modifications we are analyzing for. Okay, so you have an antibody to each one of these individually. Your band is saying, yes, DNA was associated with the specific modification. Okay, so gene A was pulled down, was immunoprecipitated with acetylated H3. So back here, if our antibody was to ACH3, then it would pull down gene A. And gene A was also associated with a methylated H3 histone. Okay. And so you can read, going across like this, which modification was associated with which gene. So this is just a yes or no assay. The RNA comes from a totally different experiment. I know I didn't make that clear in class. So RNA, we do something called RT-PCR. This is showing positive or negative for transcription, which means gene expression. And this tells you that gene E, gene D, and gene A were all being transcribed. Therefore, the modifications that they were associated with, this is getting super messy, right? Okay, so therefore the modifications that they are associated with were modifications that related to gene on. And because gene B, gene C, and gene D were missing, oh, sorry, not gene D, gene B and gene C were missing RNA, these modifications were associated with gene off. And the slide I then put up for you is said, okay, so write the histone code based on this data, and I forgot to put the RNA data there. Your questions were absolutely right. You can't do that without the RNA data, so I apologize for the confusion. So understand the kind of data I could give you is just saying, okay, yes, we detected gene A, and it was associated with this histone modification, and the RNA tells us that gene is on. So the histone code for gene A is acetylated H3, H3K4, Me3. That was its code for gene on. These genes, E and D, which are also on, have slightly different codes. And that's one of the things I want you to realize is that there's no good set of rules. I've told you that acetylation is usually associated with gene expression, but you'll see that other methylations can kind of override that. So you'll see that in some of your recitation worksheet questions. All right. I hope that is helpful in understanding these techniques. Thanks.